Okay, so hi everybody. It's a great pleasure to be here. So I'm here in Austria in the lab and in the office of a friend, and I will get back to Toronto actually next Monday. In principle, if everything goes fine. In the meantime, yeah, I'm shifted. I'm like 6 hours in the future. So for me, it's 8 o'clock, and I will finish when it's 10 o'clock in the evening for me. But that should be okay. I should not be too tired, and so in principle, everything's going to go fine. So the way it's going to work is that I'm going to share my screen, and we're going to just follow the slides along with me. Now, give me a sec. Almost there. Okay. And here we go. All right, so now I'm sharing my screen. In principle, everybody can see it. Can you guys see my screen? Very good. Yeah. Just checking. I'm still coming out loud and clear. The video is good quality. Seems it is. Okay. So once again, it's a great pleasure to be here. It's the second time I teach this course. This is my second year at the University of Toronto, and I'm really so happy to be here. I'm kind of bummed that this is online again, though, and let's see if we can get in person a little bit later in the semester. As you know, we're supposed to go back in person in February, but my advice is get ready for a different plan because we really don't know what's going to happen. There's a good possibility that this will go fully online. For us, for me, it doesn't matter so much in the sense that this class is ready to go either online or in person. So in principle, as long as everybody's safe, the class will just go naturally without too much trouble. So in terms of all these things, you don't need to worry too much about it. There would be neither penalty nor advantage for being online versus in the real world. So I'd like to first start with giving you a bit of a starter about how the class is run and how things work in BIOD25. So that's me here. I'm Guillaume Filion. I'm the professor of that class, and this is Michelle here, Michelle Harwood, who's the TA for that class. And you will interact mostly with me. There will be one class that's given by Michelle, that's the one on GWAS, but all the other classes are given by me. And yeah, for everything, I'll be really your main instructor for that class. There are one or two things where you will have to interact with Michelle, and I will tell you when we reach that point. So I'd like to introduce myself a little bit. And this is, in a nutshell, the story of my career. So I am born in Canada, but I grew up in France. So my culture is much more French than Canadian. But my name is completely Québécois, though. So people who are from Quebec, they can pronounce my name, first name and second name without problem, but everyone else has problems with it. So I started my studies in Lyon, the city of France that is around here, in 1998. So that was my first year of university. And then after this, I went for 1 year in Montreal at the University of Concordia for my third year at university. I came back to Lyon after that for a couple more years, then went to Paris to do my master's and my PhD. I graduated in 2008 with a PhD. Then I went to Amsterdam to do a postdoc. So Amsterdam is here in the north of Europe. And I stayed 4 years in Amsterdam. After that, I went to Barcelona, where I had my first PI position. And that was in 2012. So Barcelona is here in the north of Spain. And after that, I got a new PI position also with teaching at the University of Toronto starting in 2020. So that's in a nutshell, let's say the places I've been and what I've done so far. One thing is that my name is pronounced Guillaume. I know that for most of you, this is kind of unbelievable that you pronounce these letters this way, but that's me. And I curse my parents every day for that. But anyway, I survive with this. Yes, that's it. Go ahead. You have a question.
Yeah, Dr. Filion. So I'm just kind of curious. How old were you when you immigrated to France from Canada?
I was 9 months old. That just shows how much my Canadian background was developed at the time. I've lived on both sides, but mostly in Europe. Let's be honest.
Okay, cool.
It is cool. A few words about my research. So in the lab, I'm working with my PhD student at the moment. There's only one lab member. Her name is, one official lab members. They are volunteers. Her name is Manisha. And so like we have a very small size lab, and we're working on chromatin and HIV biology. So one of the questions that we ask is, what happens to HIV when it's inserted in the human genome? What happens to the expression of its genes? And for this, we're using theories that are based on chromatin, histone modifications, etc. Another branch of the lab is non-experimental. It's purely, let's say, theoretical. And I'm really interested in the modern developments of deep learning and something that's called probabilistic programming. But I won't tell you much more about this. This is just something I like to do in the lab, but we won't really touch much upon this in this class. But at the end of the semester, we'll have something about chromatin and maybe a little bit of HIV biology. So more about me is that my hobbies include blogging. I love it. I love writing more than reading, to be honest. And when I read, I really like science fiction. And with the rest of my time, I do math. So I'm a bit of a weirdo. Sometimes I just grab a piece of paper and then you'll see me just scratch stuff, be lost in my thoughts. I'm just doing some math in the background because that's what I like. Anyway, it's been like this a long time. I'm used to people finding it weird, but I still love it. And guess what? It's useful. So very briefly, the overarching goal of the course is to train you on the skills that matter. And I really mean it. So you will see that BIOD25 is not organized exactly like every class you had so far. And you'll see why as the course develops. And so now I'm going to speak a little bit about the way we're going to work online. So everything is going to be on Quarkus. And I want to make sure that everybody's on the same page. So I'm going to drive you through the different items that I've put on Quarkus so that you can orient yourself. At some point, there's going to be quite a lot of things, quite a lot of documentation on Quarkus, and you may get lost a little. So I wanted to be sure that I give you a head start right now. So the first thing is read the announcements. They will be there on top of the front page, on the home page. I think that you will also receive an email on your UTSC email whenever I post an announcement. So be sure that you read those. They will just, broadcast messages that are relevant for the organization of the class. One thing is the office hours, how to get them. It's not completely trivial because you have to actually book an appointment with me in order to make sure that you have the slot with me. Okay, so the way you do that is that there is this button here, View Course Calendar, which is on the right on Quarkus. So you need to click on that. Once you're there, that brings you to that page that looks like that. And then you need to click on Find Appointments. The office hours don't even appear here on the calendar. You can see only the stuff like the lectures and also the quizzes that are scheduled. I'm going to get back to the quizzes in a second. For now, if you want to book an appointment with me for an office hour, you just click on Find Appointment, and then they will make a couple of new slots here appear, and they're all on Thursday at 12. And this is where you can click on one and then click on Reserve, and then they'll book the appointment with me. I can't remember if there's a limit. Here it says that there are a maximum of 5 people per slot. I don't think I kept that limit. So if you want to come 20 people in exactly the same slot, it's fine with me, as long as it's fine with you. Okay, so that's not completely trivial how to book the office hour. If you don't manage, you just write me an email saying I'd like to meet you at that time, and then it's just the same. Okay, it's just to avoid having 2 people at exactly the same time in case there is too much demand for office hours. Another thing that you will see already like in an hour and a half are the weekly quizzes. So right now they're locked. Like here, this is the slide from the last year. So this is not the right date here. It should be January 12th. And the quiz is open as soon as the class is finished. So which means at 4 o'clock your time, 10 o'clock my time right now, it will open, and then you will be able to take the quiz. So there are 10 weekly quizzes, and the total percentage for the class is 15%. So each of them is like 1.5%. Now they're really low stake quizzes. Okay, so like not doing the quiz is not a huge thing for your grades. Still, 1.5 multiple times, that starts to be significant. But if you can't, then it's not a huge thing. My recommendation is still do them. You'll see it's good fun. And second, this is really the way you start to interact with the material. That's the way that you actually learn. So in my mind, they're really important, but they're still very low stake. So here it says something incomprehensible, 15% plus 3% plus 1% bonus points. What does that mean? So each quiz has actually 12 basic questions, not 10. You get max grade already if you get 10 answers right. Okay, so each question is on 0.15%. So if you get 10, that's 1.5%, but you get 12 questions. So it means that you can allow to make mistakes. Even if you make on 2 questions, you get something completely wrong, then you still get the max grade. Okay, and if you get more than 10 right answers, then you're starting to hit in the bonus points, which means that theoretically you could have more than 100% if you really score all the time the max grades. Now, it's pretty hard to get 100%, but it's still possible because overall the number of points for BIOD25 is around 105. There's also a 13th optional problem that is meant to improve your quantitative skills. So this is always going to be a math problem, and this is a random math problem. You don't always have the same question on that one. Okay, so this is just, if you feel like it, if you want to improve your math skills, you want to think about that stuff, you can try and think about the quantitative problem. And this one is on less than the others, it's on 0.1. So that's the 1% bonus point here for those who want to do the math, think in terms of quantitative skills. That is actually quite useful and quite important, I would say, in the case of genomics. All right, so that's the quizzes. Every week, except reading week and toward the end of the class, you will have a quiz to fill, and each of them has 12 plus 1 questions. You also see on Quarkus that there's a midterm, and so I already posted the content of the midterm. So one of the main concepts of BIOD25 is that there is no time limit exam, so except the quizzes. Like you have 1 week to do the quizzes, but the exams, you already know what is going to be at the midterm. This has been posted. You already know what's going to be at the final. This is already posted. So you have the whole semester to work on your midterm, on your final. There's no time limit. So that's what I meant by the skills that matter, in the sense that in the real world, there's no one that's timing you to say how good you are. Like you have a deadline and you just have to organize yourself to do the work you got to do until the deadline. And so you can have a look at the midterm if you want. In principle, you're not able to do it. Okay, you don't yet have the skills, and that's the whole point of the class, is to give you the skills so that you can do the midterm. The deadline will be after reading week. So in principle, you have the whole of reading week to do the midterm, but you don't need to spend time kind of preparing for the midterm. You need to do the midterm during reading week. I hope that's fine with everybody. If you don't understand anything on the midterm exam, don't freak out. You will have all the skills at the time when you need to do it. Okay, that's actually even better because you'll realize how much you've learned, how much you start from not being able to understand it to being able to ace it. So the final will be to write a blog post. And so again, like you won't have time limit for that. You have the whole semester to write a blog post. And the first item that needs to be done is to choose an article to write about. So kind of the major burden of BIOD25 is to write a blog post about a recent discovery, a recent study in genomics. And for those who want, and this is not obligatory, it's kind of nice, but not obligatory, it will be posted for real on the blog of BIOD25. So this is entirely up to you. If you like your work, you can publish it. You don't like it, or you're shy, or you don't want to share anything, it's also fine. Like it won't affect your grades in any way. So the first thing you need to do is to choose an article to write about. And this is already open here in the blog post module, or in the exams that are already available. So how does that work? You have to read a little bit the instructions on the assignment. And the most important part is that there is a spreadsheet with your name on it, in principle. So you'll see that after the class. You should check that. So read the instructions and click on that spreadsheet, and then it will say your name somewhere, and there'll be 2 articles next to your name. And so then it explains to you how you have to choose an article to write about. So it could be either one of these 2 articles. And so these ones are kind of your articles. No one can challenge you the right to comment any of these 2. Okay, but it doesn't have to be these 2. You have more options than that. Another option is to take one of the articles which are really at the bottom of the screen, and it says free-for-all articles. So you can't take those articles. You don't like your two articles, you think that you really hate the topics of your two articles, they were chosen at random. You hate them, no problem. You'll have plenty of other options. One of them is you go to the bottom and you see whether the list of free-for-all has anything you like. If there is, then you can choose one of those. If it doesn't have anything you like, what you can do is you can kind of steal someone else's article. But you have to ask first. You cannot really steal it. You have to ask permission, say, hey, person X, I really think the article that is in front of your name is really awesome. So if you don't really like it, I can give you mine instead if you like, or something like that. So that's another option. But anyway, bear in mind that as people choose their articles, because you got two in front of your name and you choose only one, the leftover are going to feed the free-for-all article. Okay, as people choose their article, there's going to be more options for everyone else. So if in front of your name you already see that there's something you really like, choose it now. You're going to do everyone a favor by releasing one of these two articles to put in the free-for-all articles. So that's the idea. The first thing you have to focus on for your final exam is to find an article that you feel comfortable writing about. It's got to be something that you like, that you find engaging. And again, if the two articles in front of your names don't work for you, there are other options. You'll see the detail on the page and also in the Excel sheet, in the spreadsheet. So I've uploaded the PDFs in case you can't have access. In principle, you will have access to everything from the university, but I'm aware that not everybody will be at the university. So I've uploaded the PDFs of most of the articles. Some of them are still missing. So give me 1 or 2 days. I will upload everything very soon. So in case you can't access the article from home or from wherever you are, then you will be able to access them via the page in the blog post module there. They'll be in PDF. So finally, let's talk about something that's less nice, penalties and late submissions in particular. So I know that we're all trying our best, but there's always moments when we can't, and so we turn in our work late. That happens to everybody, no big deal about this. So in that case, the rules are just to try and be fair for everybody. How does that work? So we take the example of the quiz, which is your next deadline. And here the deadline is false. It should be January 18th, actually, at 9 p.m. That's the deadline. So how does that work? If you turn in your assignment, your quiz before the deadline, there's no penalty. Obviously, you're on time. If you turn your quiz at the deadline at 9 p.m., you are late. Okay, when the clock says 9:00, in reality, it's because it's past 9:00. Okay, it's not in the process of getting to 9. It's already passed. Some people have problems understanding that. So just to be clear, if you're up and until 20:59, that's fine. But 1 second later, it's not fine anymore. Okay, so the penalty starts really at 9 sharp. And I know you find it weird that I would say something like that. But guess what? Last year, there were people arguing to death that they were not late because they submitted their quizzes in the last 15 seconds. So don't do that, please. There's no need to be like that last minute in terms of submitting your quizzes. My advice is, get a little bit of a buffer. Don't use the last seconds. And so how does that work when you're late? Well, for every period of 24 hours, you lose 20% to the final grade. Okay, so if you submit in the yellow window here, like you submit your answers, and after this, there'll be a 20% penalty applied to your results. And if you submit in this orangish window here, there'll be a 40% penalty, then a 60% penalty, then an 80% penalty. And if you submit your quiz after that, it's going to be 0. Because anyway, that's a 100% penalty. So why do I do that? Is because we need to get these damn answers at some point. So 4 days after the deadline, I know that everybody who hasn't yet submitted their quiz will get 0 anyway. So I'm licensed to give the answers to everybody. That allows me, in case someone really doesn't want to upload their quiz because, I don't know, they disappeared, and I can't get to them to say you need to upload your quiz. That's a guarantee for me that 4 days at most after the deadline, I can share the answers with you and we can talk about it. Okay, otherwise it'd be like, you'd even forget about the quiz when you get the answers. So that's the system, right? Hand in your assignments on time. If you don't, no big deal. I'm not going to be mad or something like this, but you will get penalties. So life's complicated. There's a question. That's it. Go ahead.
Yeah, sorry, Dr. Filion. So I'm just once wondering, is there like a time limit for each quiz? Like, do it to like to get them done in like an hour after we start, or?
The time limit is approximately 6 days.
Oh,
Okay,
okay.
so it will open at 4 your time, and then you will have until Tuesday 9 p.m. Every week is going to be like that. But no, you open the quiz and then you get started. You have 6 days more or less to answer that.
Oh,
You're allowed to work together. You're encouraged to work together. Please meet each other. That's my advice. Exchange tips and tricks, discuss the answers, etc. A D25 is a class where you're pretty much allowed to do almost everything together. And you're actually encouraged to do things together. You're going to learn much better if you work together than if you work alone in general. Okay, so my advice is find a friend and then start doing the quizzes with them. I know that it's going to be hard to find a friend, especially today, but from next class, we'll have breakout groups and you can find random friends. Otherwise, I know that you guys have got ways to find friends with Facebook groups, etc. So I trust you to be able to do that. Teamwork makes the dream work. Exactly. Also, it's very good for learning. You guys learn much better in team than alone. Okay, so penalties. There's one rule that's kind of important and it gets people confused a little bit. So let's explain that. The jokers. I know that life's complicated. Also, we have this horrible disease that's around and sometimes it will not be possible to hand in your stuff on time. I know that, like I submit things late, everybody does. It's okay. There's no reason to kind of, kill each other on that. So you got 2 jokers and you have to know that you've got them. So that allows you to be late 2 times in the semester. I think that's probably realistic. You have a lot of things to submit, and bear in mind that on 2 occasions you're allowed to be late, and there will be pro bono, no penalty. But you have to understand how that works. So the 2 jokers, each joker can cancel a 20% penalty. Okay, so like you can see here on this graph that's here on this schema. So if you submit it, for example, in this orange window here, if you use 2 jokers, then that cancels the 2 penalties and you have no penalty anymore. Okay, so that's as simple as that. If you had submitted here, then they would actually put you back in the yellow box. I mean, you'd have a 20% penalty because you have removed a 40% penalty with your 2 jokers. If you have 2 assignments, each with a 20% penalty, use your 2 jokers and you're fine. So the most important point now is that these jokers, they're not up to you to tell that you're using them. Okay, they're automatic and they will be applied after the semester is finished. And I will do that in a way to just maximize your grade. I will look at which are the assignments that you have submitted late. And I will say, okay, to maximize the grade, we need to negate this and that penalties. Okay, a 20% penalty on an assignment that's worth 1.5. You understand it's not the same as a 20% penalty on the final, which is worth 30. Okay, so in that case, obviously, it's more interesting to negate a penalty on the final. I think that's obvious for everybody. So the jokers are automatic. So why am I telling you that? If they're automatic, it's so that you can budget your time. Okay, you can actually rationalize the way you're going to do these things. You know that you're going to be a little bit busy in that period. No problem. Use a joker. That's fine. And then keep another one for you. You never know, for a real joker. Okay, so just think about it as adults. You have a time budget. You have a joker budget. Manage your time. I won't bother you. And then you won't write to me to say, can I please get, it was not my fault, I was late, etc. Never write to me for that because you're going to get penalty points if you do that. Okay, that's explained in the summary of the course, in the syllabus. So the reason why these jokers are here is so that we can be gentlemen, like you are late, use a joker, and that's fine. The transaction is finished. You don't need to tell me that it was complicated, that your grandmother died or something like this. I don't want to know about this. You just have your jokers, you use them. Okay, so try to understand the system. If it's not clear, you can ask me. You can come also to the office hours to ask how that works if that was not clear. But I think you must have gotten the idea by now. That's it. That's what I wanted to tell you about the organization of BIOD25. So are there any questions regarding that? Or can we already put it out of the way and start genomics? Any questions? So of course, for the questions I forgot to mention, you can type in the chat, I can read it, or you can, of course, raise your hands, Mike, and speak in front of the camera. Go ahead, test it.
Yeah, so I just had one question about the jokers. Like, does everyone get two jokers like automatically, or does anyone gotta earn it?
No, they're granted, like they're birthright jokers. You get only two, but everybody gets them. Every human gets two jokers at birth of BIOD25. That's a gift, and you're never gonna get a third one. Okay, like you cannot earn a third one. Theodore.
So say if we don't use the gift, which ultimately is a like something that we all prefer, can we use those? Can we use that 40% for increasing our grade?
The answer, it's a good question, and the answer is no, you cannot. Okay, so last year it was the case, and that created a huge amount of problems. And so I've realized that when you play sport, when you play, let's say, I don't know, basketball, if one team respects the rule, do they get 3 points? No, they just respect the rule, right? So you got the rules, you got to do whatever you want inside. I know, and I personally appreciate people who use 0 jokers. You know, for me, you'd get my respect, but no point, right? So
Soccer, we could say that it's a game. It's got rules. You can manipulate the game. You know.
sure, you totally can. But let's say just respecting the rules doesn't get you points. It's just respecting the rules. That's why it is.
Thank you.
Any more questions? Tasneem.
Hi, Professor. How are you?
Good, thank you.
I just wanted to ask, so you said that we are encouraged to collaborate together.
Yes.
So can we do that during the midterm and the final too?
Absolutely.
Okay, and the quizzes are open book, right? So we can see our lecture notes and everything.
You got 6 days. You can do whatever you want. You can use the internet, you can use Google, you can use anything you like.
Oh, thank you.
It's the real world, you know, like what I mean by the skills that matter is I'm trying to get as close as possible to the real world. In the real world, there's no one to cut your internet or to say that you have only an hour to do a task or something like this. In the real world, you have everything to solve your task, but the tasks are difficult. Okay.
Okay, thank
so,
you.
A pleasure. Just to follow up on that, why are you allowed to work together on the midterm and on the final? Why is that not a problem? It's because you each have a different midterm. Okay, the questions are the same, but the midterm is actually working on the computer. And so you will each have a different file to work with. And so that means that I can give you the same questions, but anyway, your answers are different. So you can actually use each other's knowledge, each other's understanding of the class so far, which is what I want. I want you to understand well what happens. But anyway, you need to apply it in a different way to your problem because you don't have exactly the same problem. In the case of the final, you don't have a proper final exam. It's a blog post. And so you will each have a different article to write about. And I really encourage you to read each other's article. Actually, that's a very good thing to get ideas, to stimulate your creativity, etc. But that won't really give you points, you know, to look at someone else's article. You will still need to find a way to talk about your stuff. So that's the philosophy of BIOD25, is that the exams are unlimited time, they're free, but they're personalized. Each gets a slightly different variation of the exam so that it's to promote collaboration instead of saying that, you know, collaborating is cheating. No, in BIOD25, collaborating is normal. That's even encouraged. And for the quizzes, you each have the same questions. And I'm totally aware that some of you could, like, you know, just look at your neighbor and say, yeah, just give me the answers and I'll just feel that. It's fine with me. You know, if you don't want to learn anything through the quizzes, it's okay. It's anyway a very, very small budget in terms of points. So you're kind of missing an opportunity to learn in reality. And on top of it, you have to trust your friend that they're better than you. It's not always the case. So that's the reasons. Any more questions?
Thank you, Professor.
Pleasure. What exactly is the whodunit? Very good question. So that's the exception to the philosophy of BIOD25 in the sense that this one you will have less time to do it. So it's going to be a kind of crime, mystery, murder story that you will have to solve using genetics, a little bit like CSI. And this one, I will release it only 1 month kind of before the deadline. And so it means that it will be just after we have the class that gives you the skills to do that. I will prepare. So the reason why I'm not opening it right now is because it's not ready. That's the one that's the most difficult to give you each a different problem. And so I need to work on that a little bit. And so it's going to be just like, you know, solving murder mysteries, etc., using genomics. Alrighty, I think there's no more questions, so let's get started with proper genomics. We have a lot to cover. I'll make a little break, let's say at 3 o'clock for you. Like we'll have a short break, but today there's a lot of material to cover, so I'll try to make the break relatively short. Let's get started. So today we're going to talk about the human genome and the SARS-CoV-2 genome. So what makes us Homo sapiens? Well, basically, it's our genome. The way you put it, you could say it's your personality, it's your thing, it's your values. In the end, that's biology. Homo sapiens doesn't say anything about human at any other level than biological. So what makes you human is your genome, our genome. Our genome consists of 22 autosomes, plus the X and the Y chromosomes, plus the mitochondrial genome that everybody forgets all the time. This represents 2 times 3.2 billion nucleotides of DNA that make each of us Homo sapiens. So that's here a carrier type. I'm sure that you guys have seen that before. And you remember that the chromosomes are in pairs, one from mom, one from dad. And then you got the XX for women and the XY for men. And what's not represented here because it's way too small, it's invisible, is also the DNA that is in the mitochondria. So the reason why we know what's inside the genome of the human species is because it's been sequenced. And of course, for you, I think that most of you were born when that was already natural. So a participant requests that live transcription be enabled. Of course, there's no problem. One thing that I have to say is that the lectures are recorded, and I'm going to post on Quarkus the version that has the subtitles. Okay, and usually the subtitles on the recorded version are a little bit better than the one that are produced now. Zoom is not always very accurate. Actually, if you read the subtitles, sometimes it's really hilarious. Anyway, I know that for some of you it's difficult to understand. If I don't speak clearly enough, if my English is too hard to understand, if I speak too fast, or just if I mumble or whatever, if you don't understand me, you can just tell me, and I'll repeat whatever I say, or I'll speak slower. There's no problem. Okay, so just if there's anything unclear, you tell me. So the reason why we know what's inside the genome is because it's been sequenced, and most of you are kind of born after the, or at least at the same time when the genome was sequenced, but you live in a world where the internet has existed all the time for you, and where genomes were sequenced all the time. You can remember a time that was before, just because of your age. But in my case, that happened precisely when I was a student, all these things. So I can clearly remember a world before the genomes were sequenced and what happened in these historical years around 2000 where the human genome was sequenced. So that's here a very brief outline of the Human Genome Project. And you can see that the years, well, they start kind of 1984, but not much happened at that time. It really started in 1990, and you can see that this stops in around 2001. So there's a lot of things on this graph, and the thing that matter are in green here, that's the human genome that are there. But what they want to show on this graph is that it's not the first genome that was sequenced. There were much smaller genomes that were sequenced before, like the one of the yeast, Saccharomyces cerevisiae, C. elegans, etc. The flu E. coli that were sequenced before the human genome. But in terms of the magnitude of the project, the Human Genome Project was really something that was an unprecedented scale. And there will never be in genomics a project that demanded that much work as sequencing the human genome. So that's why I want to talk today about this, because it's by far the most important genome that we've sequenced. Well, almost the most important genome that we sequenced. We'll see that to the end. So before 1990, there was a little bit of genetic analyses that were done, and you can see that there are genetic maps, physical maps, cDNA sequencing, genomic sequencing. And one funny thing is that around 1999, only 15% of the genome had been sequenced, and around 2001, they announced that it was finished. So in reality, the sequencing was a relatively short window of 2 years, but there was a lot of preliminary work that was done from 1990 to 1999. And there was also an event that precipitated the end of the human genome. It was actually supposed to be finished around 2005, but they accelerated in the end a lot in order to meet a certain deadline. So the completion of the human genome was officially 2 times. The first, well, it was finished on the 26th of June 2000, and they called it the RAF draft. And so there was a press conference with the president of the time, Bill Clinton, a person called Craig Venter that you can see here, who was the head of a company that is called Celera Corporation, and a person who's called Francis Collins, who was the head of the National Human Genome Research Institute. So in 2000, they announced that the human genome was kind of finished, at least in draft form. And again, another time, they announced that it was finished in 2003. So they did this. The target date was to match approximately 50 years after the discovery of the double helix by Watson and Crick. That was on the 25th of April, 1953. Yes, Yibing, you have a question.
Hi, Professor. If I'm not remember it wrong, like I learned from another class that like the human genome we sequence, the one that we completed in 2003 is kind of still have gaps, like not really that complete. Is that true? Like,
yes, that is true. And even today, the human genome is not completely finished. I'm going to give you more detail in a couple of slides.
okay.
So back then in 2000, it was really not finished. In 2003, it was kind of finished, but not completely. So why? And also, why did I say that they accelerated? It's because everybody was afraid of the Celera Corporation. They started much later than the public project. There was a multi-country consortium. Okay, so like the leaders of this were the American government, but there were also the UK government and other countries in the world that were doing this sequencing project. The problem was that this private company led by Craig Venter was actually going faster than the public project. And at the time, it was still okay to patent genes. So people got really scared at the time that this guy would actually patent and own the human genome. So this is why there were some agreements, in particular involving the president of the US, to say, no, let's say that this is a, that's a tie. You both arrived at exactly the same time on the 26th of June 2000. And that also means that because the public project is public, then nobody is allowed to patent human genes. A couple of years later, anyway, lawyers kind of established that regardless of what happened in 2000, human genes are human patrimony. You can't own them, you can't patent them. So it's not completely true because the laws are not the same in Europe and in the US. So it's complicated, but basically that was political. The decision to say it's finished in 2000, it was just because that was a good excuse to say they both finished at the same time and there's no clear winner. In reality, it's only in 2003 that really you could start to use the human genome for proper experiments, because before that, it was really drafty. So let's see a little bit the reason why the Celera approach was getting faster than the public domain, the public consortium. So there were two strategies. The one that we're going to describe today is the one of the public consortium, and that's kind of the major endeavor. That's the one that took 10 years. The one of Celera Genomics, or the Celera Corporation, took much less time. It was much more cost effective. And I'm going to describe it rapidly today, but there's going to be a lecture actually on that method in particular. So the method of the public consortium was called mapped clones, and that works like that. The first part of the project, the one that took 8 years from 1990 to 1998, was to make landmark genetic maps using recombination markers. I'll explain that in a second. Then you make physical maps of overlapping clones. So they really obtain maps like, in 1D with actual coordinates of different pieces of sequences. They wanted to make sure that they knew that some key, let's say, milestone sequences were had known location in the human genome in order to arrange everything in an easier way. Then, and only then, they selected clones to sequence. So that's only at the end of the project, starting from 1998, something like that. And then they assembled everything to make these genome reconstructions that we have. The method on the right by the Celera Corporation is kind of different. That's called whole genome shotgun sequencing, or double barrel shotgun sequencing, because they were sequencing the pairs. So the first step here is to shotgun sequence random short inserts. So they just shatter the genome and then sequence things at random, completely at random. based on what they sequence, just like a puzzle. You know, when you have a sequence that you've seen, you say, oh, these two, I can stitch them together because I've seen that sequence before. So you can start to stitch the reads a little bit like that. So they make small reads here, longer reads in which they have only the ends, but not the middle, because it's harder to sequence the middle. And based on that, they take the orange reads with this stitching method to create what are called contigs. So that sequences that are contiguous, that's where the word contig is coming from. And with the green reads that have, you know, this kind of, that sequence only the ends of relatively long fragments, then they can know where the contigs go together. So they can arrange them in scaffolds. And that method here is much more risky than the one on the left, but it's much cheaper and much faster. So with that method of shotgun sequencing, they were going much faster than the public consortium. Like I said, we're going to speak mostly about the method of the public consortium. Yes, Theodore.
So for the shotgun sequencing, like if it's faster, wouldn't it be more prone to errors?
Yeah. Yeah, of course. There's no free lunch. Like it's more prone to errors, but on the other hand, you can compensate by just sequencing more. Like the costs are just so much in favor of that method that it's still the way we do it. Like the only genome that we sequenced with the old-fashioned way is the human genome. No one will do that again. It was way too much money, and nowadays you can use the shotgun method. You just sequence more. Sequencing has become cheap anyway, and then you can compensate for the problems. That's a very good point. I'll explain in a second exactly what are the problems with the shotgun approach. So anyway, we go back to the traditional approach, the one that was used actually only for the Human Genome Project. So how did it work? Remember these first 8 years where nothing was happening? In reality, they were doing just basic genetics. And I know that you guys must know what I mean, because this is a prerequisite for genomics, is that you're comfortable with these notions of crossing over, centimorgans, etc. Don't be afraid, we don't do that, but it's just so that you understand how that works. So they were measuring crossing overs from large families. You got the grandparents there, the parents, and they're like 10 plus children. So they could measure relatively accurately the crossing overs between plenty of regions in the human genome. And so they were measuring crossing over between alleles that can be detected by PCR. And those are microsatellites. That's something that we'll see next class. And they could order them on the chromosome. The same way as you guys have made these exercises, you've gone through these exercises when you say allele A, here this marker A is on the left of B, which is on the left of C, etc. You can order them relative to each other. So that's what they were doing for many years. They were ordering all the markers that they could lay their hands on to know where they are relative to each other. But that takes time. It takes a huge amount of time because you need to get these people to, to find these families that have so many children that you can map everything very accurately. But with that, they got markers with a known sequence, and the relative location with each other was known. And that's the point. Whenever they would sequence this in their clones, they know, oh, okay, this is on the left of that because we've sequenced that before during the first 8 years. So the next thing that they were doing is to actually sequence the DNA starting from 98, 99, if you remember. And they were sequencing what are called back libraries, bacteria, artificial chromosomes. So what's a back? It's just a big plasmid. A plasmid is a circular piece of DNA that grows in the bacteria. So you take a plasmid here in a sense, and then you put a huge chunk of the human genome. When I say huge, it's 100 kb, 100,000 base pairs. And so you put it inside, so it gives you kind of a ginormous plasmid here that you can put inside the bacteria, and then while they grow, they replicate that plasmid because they believe it's their genome. So that's a back, just a big plasmid really. So each of these big plasmid contains 100 kb of human genome, and you just need to sequence the 100 kb. Then it gives you a piece of the human genome that's approximately 100 kb. So they were creating these back clones, putting them on agar plates there, picking the agar plates, and then preparing the DNA that gives you a large amount of DNA from the human genome. Makes it relatively easy to sequence. So how did they sequence these things? It looked like that, a little bit like these old-fashioned science fiction movies, things that wanted to be futuristic but were totally not. So these are automated sequencing centers in the 90s. Like these things are mid-90s or end of the 90s, both of them. And you can see that, well, there are a couple of computers, etc., and these machines which are Sanger sequencers. So automated sequencing was not really a big thing then. So they really had to invest a lot in buying these machines and making these, these ways to automatically sequence the back clones that they were having. And they always used the same method. There were known other methods to sequence DNA, but the human genome has been sequenced almost exclusively with one method, and it's called the Sanger sequencing method from Fred Sanger, who got the Nobel Prize for inventing the method. Okay, so almost all the human genome, every nucleotide in it, has been sequenced with that method. It's a very important method. I'm going to describe it to you a little bit today, and next time we're going to spend more time on it because this is really still the standard. That's the way that pretty much most of the sequencing methods are based on. So the Sanger method works a little bit like this. That's not a super good sketch of the Sanger method, but that's the best I could find on the internet. So here, that's the molecule you want to sequence. So you know the sequence here for the sake of the argument, for the sake of this example, but you have to imagine that really you don't know the sequence you want to sequence. Otherwise, you'll not be sequencing it. So it goes from 5' here, that's only one strand that is shown here, to 3' with a GACT, etc. And the gist of this method is that you're using a polymerase to elongate that molecule here. What is not shown and what makes this sketch not so good is that there must be a reverse complement strand to that, and that's the one that's used as a template. And the template is used to produce that strand here when there's a polymerase that elongates the nucleotides on that template. And the most important part of the Sanger method is that among the normal nucleotides, the DNTPs like A, C, G, and T, there are the dideoxy nucleotides, the DDNTPs. And these ones are blockers. So which means that once you incorporate one of them, you cannot anymore elongate the DNA. That's finished. And really the genius idea of Sanger was to combine these with different colors. So all the blocking Gs are in yellow, all the blocking Cs are in blue, all the blocking As are in green, all the blocking Ts are in red. So which means that if you just spike in a little bit of these blocking nucleotides, let's say one in 100 or something like this, you will be able to elongate the DNA normally, but then out of the blue, at random, you're going to incorporate one of these blocking nucleotides, and then that's finished. You can't elongate anymore. So in this sketch, it goes from the top, it goes down. So if the first blocking nucleotide you use is G, if it's the first, it's got to be a G because you need to incorporate a G, and that's finished. You have only one nucleotide and you're done. Or you got lucky the first time, you have this non-blocking nucleotide, and then you incorporate the non-blocking nucleotide. It's going to be an A because you're producing that molecule. So you got an A here, but the molecule is 2 nucleotides now. It's a bit longer. Okay, and so on. Like you always start from the top, and as soon as you hit one of these blocking nucleotides, the molecule is finished. You can't elongate it anymore. But because these blocking nucleotides have different colors, it also means that every time you finish on a G, the molecule is yellow. Okay. Every time you finish on an A, the molecule is green, etc. So that allows you to know just by the color of the molecule, which was the last nucleotide that was incorporated. And the final trick that they use is that once you've done that, a couple of times, that you elongate these products of different length, but whenever they stop, then they get a certain color. And then you do an electrophoresis that separates them on the sides. And the molecules that are the shortest one, the ones that contain only a G here, they are small, so they migrate fast. And the molecules that were blocked only toward the end here, they're kind of heavy because they have a lot of nucleotides, and so they migrate much slower. So that gives you here a typical electrophoregram in which each position gives you a size, that's a number of nucleotides, and each color gives you the nucleotide that was the last one that was added. So you can actually read the sequence just like that, GAC. I mean, you read like yellow, green, blue, red, etc. But that's like yellow, green, blue, red, etc. So that's GACTG. So you can just read the sequence directly on the electrophoregram. That makes it a really simple method in order to decode DNA, to sequence it directly. Okay, so that's kind of a complicated scheme because you have like, multiple methods that you got to be comfortable with. And like I said, we're going to spend more time next week to really understand that method in depth, because that's really the most important one to understand everything that came after that in terms of high throughput sequencing and all. So now is a good time to have a little break. So I'll take questions if there are any, and if not, then we're going to take a 5-minute break. Are there any questions? Doesn't look like it. Okay, so I'm going to pause the recording for 5 minutes, grab a coffee, do whatever you want. See you at 5 past. Okay, welcome back if you're watching this online. So there's a question. Professor, sorry if this is a silly question, but we talked about 2 strategies, the cellular corporation strategy and the public consortium one. So is the public consortium the same as the National Human Genome Research Institute? Yes, kind of. So this National Human Genome Research Institute is actually an American, like US institution, but the public consortium was actually a multi-country consortium. Okay, but you can consider it's almost the same thing. It doesn't matter so much. What you have to understand is that there were really 2 competing projects, one from a company that was trying, sorry, that was trying to do this for profit, and one that was academic, and that was actually involving several countries working together, but that didn't really have any notion of profit. They were doing this because they thought that it could be a good thing to have in order to work on human species, basically. Okay, so we're now going to go back to the presentation where we left it. So let's see. A little bit before that. Okay, so the assembly. So now we've seen how the public consortium was sequencing these pieces that were in back clones, like bacterial artificial chromosomes, and that allowed them to stitch them together whenever they were finding the sequences that were overlapping. Okay, you can imagine that you're going through your, yes, the recording is on, I think so. It's kind of hard to know because my screen is really tiny here. When I'm in Canada, it's going to be a little bit better. So yeah, I think the recording is on. So now if you imagine that they were sequencing the back clones, so they would kind of start on both ends and then like use this Sanger method to kind of creep inside the back clone. And so once they have the whole back clone, then they need to find another back clone that's next to it in order to stitch them together. So these things here in blue, they correspond completely sequenced back clones. And when they have a lot of sequence overlap, you can say, okay, this one goes like that, the next goes there, etc. But sometimes you get gaps, like you never sequenced anything here, for instance. So what do? You can't really continue this method. And in that case, they could really use the method that they had developed in the first 8 years, which is to say, well, it doesn't matter for sure exactly what their distance is. We know their relative position because we have these markers that we've mapped through recombination, through centimorgans, like the way you've done it in other courses. So we know that this piece here is on the left, and it's approximately so many nucleotides from that piece here on the right. And we know this because we've got these markers that we keep sequencing all the time in our back clones. And so that's something that the other method cannot do. Whenever you have a gap like that, you've got really no idea where they go. You cannot even know on which chromosome that they go. And that's the big difference between these two strategies. Like the public method had a safety net. They always knew at least the relative position on every back clone. And for sure, they always knew the chromosome. In the case of the cellular genomics approach with shotgun sequencing, when they didn't get any clue here, then they could not know at all where that piece would go. So they got contigs, and because they had this strategy of being able to know where the contigs go, they could scaffold them. So a scaffold is a continuation of contigs, but you've got gaps in between. You don't know for sure exactly what's the distance here between the contig on the left and the contig on the right. So overlapping reads are stitched together to form so-called contigs, comes from contiguous, and consecutive contigs can sometimes be arranged into scaffolds when their physical location is known. And that was the case for almost all the contigs of the public consortium, but not so for the cellular genomics. The thing is that the cellular genomics were using the public consortium because guess what? The public consortium were releasing their data. It was a public project. So cellular could have their cake and eat it because they could actually use all the work that was input by the public consortium to know these markers. So in reality, they also had a safety net, but they just didn't produce it themselves. So at the completion of the human genome around 2000 came this wonderful tool that was called the genome browser. And that really marked the genomic era, the fact that we can finally not just think about genomes, but we can visualize them. We can have a feel for what they look like. And I'd like to spend the rest of that presentation of the human genome through the UCSC genome browser. That's something that you can do yourself. It's online, it's for free. And so if you just Google UCSC genome browser, you'll get to a page that looks like that. And so I've chosen to start the journey here on chromosome 17 at the location indicated by this red bar here. And that's a gene that's called CSH1. I know almost nothing about that gene. That's not a gene that's very interesting in many ways. But it was the first human gene to be sequenced. So in 1977, it's because it's very small, also because it was relevant. It's a hormone, I think. It was the first gene to be sequenced. So the journey of the human genome sequencing actually starts here, somewhere on chromosome 17. And what you see here is a snapshot of that gene. So let's see a little bit what the features are of that gene using the genome browser. So you've got here the indication, that's the whole chromosome 17 with a banding pattern that represents a little bit how the chromosome looks like when you have a karyotype, when you have a slide here. And here is really the place where we're zooming in, and it's a tiny place in a very large chromosome. The genome is actually a large place. And you can see here something that looks like this. These boxes here are coding exons. So part of the mRNA, of course, the mRNA produces, like a non-mature or premature transcript that will be mature, that will be spliced. And what remains are the exons. They're conserved, which means that between species, when you look at the sequence here, they're quite similar, even if the species are different. And exons correspond to approximately 3% of the genome. That's a tiny portion, You also have the fact that there's not just one line representing the gene CSH1, you got several, if you can see here, and they don't look exactly the same. And in particular, you can see that this here is a box on that line, and on the other ones, it's just a thin line that's there. So these are alternative transcripts. There's one gene, but it produces multiple RNAs. So these are different mRNAs from the same gene. That's a concept that I'm sure you already have, that you can produce through alternative transcription, alternative splicing, more than just one RNA per gene. You have non-coding exons, so they have a box, but they're non-coding. So how can an exon be non-coding? Well, it's quite simple. Transcription starts always on an exon. You don't remove the beginning of the RNA. And you don't start the transcription at the same place where the translation starts. You need an ATG for that. So the beginning here is always non-coding. That's called 5' UTR for 5' untranslated. So you can see that the box is a little bit thinner here to indicate that that's exon, but that's not translated. And the same at the end, you've got a 3' UTR, 3' untranslated. And you have introns that are indicated here by just this line. So that's the part that are removed from the gene by the splicing machinery. You also have arrows to indicate which direction the gene is transcribed. So that already gives you a little bit of an idea of how the genes look like. Yeah, quite simple. Like they're a succession of exons and introns, and the same gene can actually produce multiple versions of the RNA through alternative splicing, alternative transcription. You can see this track here on the UCSC genome browser, which is the conservation. And so the higher it is, it means the more the sequence looks the same in other species. And that's pretty much spot on with the exons, as you can see. Like the introns are not conserved. And the reason is because it doesn't matter what you put in the introns. It's not coding. It doesn't really do anything most of the time. And so whatever sequence you put there is just fine. Yes, Theodore.
Sorry if I'm bombarding you with questions. Just
Fine.
so the first line, the CSH1, the first line, if you see, it shows the arrows, there's pointing left, and on the other side, it's pointing right. And you said that's the way they're being translated or transcribed. Can't remember what you used.
Transcribe. So here, like you can see on the introns, the arrows are going this way from right to left. That's the gene is transcribed from right to left. And here, these little arrows that go here, they just mean that the gene continues a little bit more on the right. It doesn't mean that anything is going that direction. It's just because my zoom was a little bit too narrow.
No. But does that have any like significant importance that we need to know? Or
yeah, the centromere is here. Look at the chromosome. Okay, so that kind of funny double red arrow here, that's the centromere. And so we're here on this little red mark, which means that when you look at the chromosome this way, then the gene is oriented like that. The polymerase starts on the right and goes to the left. It's not the other way around. So that's a landmark that's absolute. Okay, so like the gene is oriented like this and not like that. At least if you fix the centromere and the left arm and the right arm the way they're represented. So we can now focus a little bit on the architecture of genes. Like the example of that gene is fairly standard. There's nothing special, nothing atypical about that gene. So all the genes look a little bit the same. They have a promoter, also known as a transcription start site. And this is upstream of the transcript. This is where the transcription begins. Okay, that's the definition of a promoter. They have enhancers as well. So that's the ones that are indicated in yellow here. Sorry that I did not mention it before, but most of the time we write the genes from left to right because like in European alphabets, we read from left to right. And so like that sketch here, the gene has been in kind of the canonical orientation, which is the promoter, and the beginning of the gene is on the left and the end of the gene is on the right. So the promoter is here in yellow. You have an enhancer or a silencer here in yellow, which can be further downstream or upstream from the gene. So enhancers activate transcription from a distance, and they can be pretty far, up to millions of base pairs away, either downstream of the gene or upstream of the gene. Finding enhancers is hard because they are poorly conserved, but it is clear that there are much more enhancers than promoters. There are well above 100,000 enhancers in total, for instance. So that's the typical structure of a gene here with a promoter, a couple of enhancers, and then you can see that there's the 5' UTR here that I mentioned, the 3' UTR, a couple of exons that are indicated in red, a couple of introns that are indicated in gray. The introns are removed during transcription or shortly after. And then that creates a mature transcript, which has a cap here and a poly A tail that are added after the transcription. And all the introns are removed, so that makes only a coding sequence that creates a protein that's folded in space. So that's the typical structure of a gene in the human genome, but also in most genomes. So what about introns? Most introns are short. That's the thing that even people who work on genomics are not aware of, is that most introns are actually very short. But several introns are gigantic, and they make up for a very large intron coverage in the genome. So that's here a histogram representation of the sizes of the introns, and you can see the sizes that are there. So really most of the introns are less than 500 base pairs. That's not very big. For a genome that's 3.2 billion nucleotides, 500 is really, really tiny. And you can see that the very short introns are very frequent. But of course, because they're very short, they don't cover a lot of the genome. What covers a lot are these absolutely enormous introns that are more than 20,000 nucleotides here. And so when you sum up the introns, which are mostly coming in the form of few extremely large introns, then that's approximately 1/3 of the human genome. So approximately 1/3 of the human genome is transcribed, a little bit more, and almost everything is not interesting for protein production because it's removed. Okay, when you look at the pie chart here, there's a huge fraction that goes inside introns, about 1/3 of the genome, and exons, remember, it's 3%, almost nothing. So some people have questioned what's the efficiency of such a system if you produce so many RNA nucleotides that you instantly remove and then throw away. What's the point? And it's not completely clear. There are some arguments about that system, whether it's adaptive at all. But for sure, you have to remember that human genomes, I mean, genomes in general, were not really optimized. They evolved. Right? They were not designed by an engineer. So you have to imagine that this thing has been going on for approximately 3 billion years. And if introns were there, you can't get rid of them at some point. They're there, and you have to make an organism that has them. Anyway, that's the way it is. Most of the introns are small, but some of them are so big that in the end, the coverage in terms of introns is really big in the human genome. So now we start to zoom out a little bit. There is this zoom button that's in the UCSC genome browser. You got the zoom in here, you can zoom in, or you can also zoom out. And here I clicked on a 10-fold zoom out. So I'm just going away from that gene, still our favorite gene that is there. So what do we see here? We still see the conservation here that corresponds to the places where the exons are. We start to see a couple additional features when we start to zoom out away from the gene. So these things that are indicated in yellow are the regulatory sequences. There are the enhancers that I mentioned in 2 slides ago. So enhancers activate genes. They're non-coding. They're not conserved. As you can see here, the conservation score here are pretty low. Okay, so enhancers are important, and yet they're not very well conserved. And it's a bit of a mystery how that is possible. They correspond roughly to 6% of the human genome, twice the size of the exon. So there's more regulatory sequences than what is actually used for producing proteins. We also have an interesting track here that starts to have some activity, and that's the repetitive sequences, repeat masker. So that represents transposons, integrated viruses. It is not conserved. As you can see here, there's no conservation whenever there's a repeat. And they correspond to approximately 50% of the genome. That's right, about half of the genome is on that track here, on the repetitive sequence track. We also see things that are annotated as SNPs, single nucleotide polymorphisms. And that's the pieces of the sequence where we are different. When we sequence your genome and mine, then we're not going to find exactly the same. So everywhere else is the same. Like we can just look up the sequences from the internet, but on these regions that are indicated by these little notches there, then we don't know exactly what you have and what I have because we're different on these regions. That's the diversity, that's the SNPs. And there's one SNP every 1,000 nucleotides, more or less. Okay, every time you sequence 1,000 nucleotides of human genome, well, there's one of these nucleotides that's not the same for everybody on the planet. That's diversity. So we start to see that it appears quite a lot here when we zoom out. There's quite a lot of diversity. There's 1 per 1,000 nucleotides. So repeats, we said 50% of the human genome. So actually, we're much more repeats than we're ourselves, in the sense that we're only 3% of exons, which produce our hair, our skin, and our teeth. Everything that we are is actually the product of proteins that are enzymes or that are even constitutive proteins. And that's only 3% of our genome. 50% of our genome is repeats. What are they? What are repeats? So there are 4 main categories of repeats in the human genome: the lines, the signs, the retroviruses, and the transposon fossils. So the line stands for long interspersed nuclear elements, and signs stands for short interspersed nuclear elements. It's a horrible name, doesn't mean anything. This back from a time when people could not even sequence the genome. So they gave a name that they thought was funny at the time, but that doesn't mean anything. So it's okay if you don't understand what it means. I don't understand it either. So lines, let's look at it. They look like this with ORF1 and ORF2. that are there. So it's a very simple structure. They're kind of small, 6 to 8 kb, so that's fairly small, but there's really a lot of them, like 85,000 of them in our genome. So just that, okay, just this 8 kb has been repeated 85,000 times, and that covers approximately 21% of our genome. So we mostly that. So in terms of ourselves, what we are in our genome, it's mostly that stuff that has been copied 85,000 times. The signs here, I've got a completely different structure. I'm going to speak a bit more about this in a second with an L and R elements. They're much shorter, 300 base pairs, but there's much more of them, like 1.5 million. So that sign sequence or the allele sequence is really present in many, many copies, but it's short. So the coverage is only 13% compared to lines. Retroviruses, are there really viruses in our genome? Yeah, 8%. So our ancestors got infected by a virus like millions of years ago. They went into the germline, and guess what? This thing is still here. And is it completely dead? Not always. Some of them can even, sometimes they're just asleep. They can wake up, and then you get a new infection out of someone that was infected millions of years ago. How about that? That's 8% of our genome. Most of it is completely inert, though. The number of active retroviruses in our genome that we got by descent, just for being human, is very small. We're talking about, a handful. So you don't need to freak out about this. You're safe from that. You don't need to worry. But there's still a large part of the genome, 8% more than exons. Then you got transposon fossils. It's not really clear how these things got there. They range from different sizes. It's more of a mixed bag, these ones, because they have different structures. They have different phylogenetic origins, and some of them can be really, really old, being in our genome for maybe 1 billion years or something like that. And they're kind of not too big, only 3% of the genome, also their number is small because they don't really make copies of themselves. They just jump around, these transposons. It's kind of a weird thing. So that's it. Most of our genome corresponds to that. It's not even ours. It doesn't even contribute very much to who we are. It's kind of weird. We're not really ourselves in a sense from the biological perspective. So line 1 is a very interesting piece to think about. So that's the structure of line 1 here with ORF1 and ORF2. You can look up the details of what's in there. It's from Wikipedia. And you got the sign here, which is called ALU in the case of the human genome. And the point is that the signs are actually coming from the lines. Yes, that's it.
Oh, sorry. So, hey, Dr. Filion, so can you just go back to the last slide one more time? Yeah, so on this slide, I can see like in terms of the coverage, like the numbers only add up to like near like 50. So like what are the other proportions of like our gene coverage?
The remaining 50%, you mean?
Yeah.
Yeah, it's a little bit less than 50%. So if you look at, for example, that introns plus exon, that's what's transcribed. What's transcribed are more or less genes. And you see that makes like, I don't know, roughly 40%. So now we're missing only 10%. Like you got 50% of repeats, 40% of genes, more or less. And then the remaining 10%, there are structural features like centromeres, telomeres, etc. That adds up to quite a lot, roughly speaking.
Okay, thank you so much.
Pleasure. So the lines here, it's a retrotransposon, and the sign is just a very weird thing. And the point is that the lines are creating their 21%, but they're also creating the signs. So how does that work? Lines create two proteins. So it kind of were not really inspired when they give the name. They call it ORF1 and ORF2 for open reading frame 1 and open reading frame 2. An open reading frame is just a gene, a protein coding gene. And so what are these ORF1 and ORF2? So in red here, you can see the RNA of the line here, which is this retrotransposon. And the RNA goes to the cytoplasm. It is translated, produces ORF1 and ORF2, and these proteins instantly bind the RNA that was used to produce them. And ORF1 stabilizes the RNA, and ORF2 is a reverse transcriptase. So this RNA here in red is reverse transcribed. It means, we'll see this a bit more in detail in later classes. It produces a DNA from an RNA. That's what means reverse transcription. And it is incorporated in the genome. It is copy pasted from the original location, sorry about this, of the line 1 to a new location. Okay, like the line 1 is transcribed, goes to the cytoplasm, reverse transcribed into DNA, put back again in the genome at another location. So that's +1 copy every time that happens. Most of the time, it is the line 1 RNA here that is reverse transcribed, but sometimes it is the unrelated, completely unrelated 7SL RNA that sneaks in. And so that RNA just has one thing that it's good at, is to go inside that complex, kick out the one that corresponds to line, and be reverse transcribed instead. And just because it's good at doing that, then it made this 1.3 million copies of itself in DNA form. Okay, that was an RNA that was used for something completely different, got reverse transcribed by accident through this line 1 reverse transcriptase here, and that's the RNA that went back to the genome instead of line 1. So in reality, line 1 is responsible for the 21% that it produces copies of itself, plus all the copies of the signs, because the signs are produced from the reverse transcriptase of the line. So here, that's the burden of the line in our genome. That's, let's say, 45% of the genome is just coming from that single element that's called line, because they produce both lines and signs. So that's a very, very, dominating phenomenon happening in our genome, and yet it's otherwise completely invisible. So we zoom out further and we keep pressing the zoom out button, and you can see now that when we do that multiple times, we can have a relatively large window that corresponds to 1/3, 1/4 of the chromosome here. And that's the way it looks. We got plenty of genes that are lined up in that view here. And what else do we see? So we can see, for example, that particular region where there's no gene, there's a gene desert. And that corresponds to this remaining 10% where we don't really know what is there. It's neither gene nor repeats. Like, these are sometimes mysterious places. This is rich in repetitive sequences, and this is not served. Some regions of the genome have very few genes. They're called gene deserts. It's not always clear why that is. You can also start to see something that we didn't see a lot before, and these are the SNPs that are highlighted in green with the green tick marks, or meme alleles. So these are disease-causing variants. Remember that we have SNPs. These are the places where we're not all identical. And some of them are actually causing diseases, phenotypes. So these are the so-called genetic diseases, of course. And there are approximately 6,800 known mutations that cause a phenotype, that cause a disease in human. That's a tiny minority. You have to compare that to approximately 3 million SNPs, where we're different. So most of the SNPs do nothing at all, like they're just neutral mutation, but there's a good 6,000-7,000 of them that actually cause mutations that, sadly enough, cause genetic diseases. So when you think of variation in the human genome, you don't have to think of necessarily causing phenotypes. Most of it is completely invisible, like you're normal in any sense of the term, whatever SNP you have. It's only in one of those very, very few SNPs that things start to go wrong. So now it's time to speak a little bit about the genes. So we talked a lot about the genome. So what about the genes? So this slide is just to remind you that you've got to be top of your game for the genetic code and translation, because we're not going to review that very much in this course. So transcription happens in the nucleus. Of course, the RNA is then exported to the cytoplasm, where translation happens. Translation happens in the cytoplasm. Ribosomes produce a protein from the mRNA and from tRNAs that are loaded with amino acids. I'm sure you guys have seen that multiple times already. The genetic code establishes a correspondence between codons and amino acids. And I expect that you guys are familiar with these concepts in this class. We're not going to review that very much. That slide here is just for you to get your things together when you go through the slides, but we're not going to spend much time on it. So what about the human genes now? 1/3 of the genes are involved in signaling, okay? So here you've got the pie chart that corresponds to the functions of the human genes. I want to say genes in this context, I mean protein coding genes. So there is approximately 23.6% that is unclassified. 1/4 of our genes, we don't really know what they do. Okay, about 1/4, we know that they're there. We know they code a protein, but we've got no idea what that protein does. So they end up in that category here, which is unclassified. The other ones, we don't know for sure, but we've got an idea. And when you sum up the receptors, the things that are on the surface of the cells, for instance, the transcription factors, the genes that regulate other genes, the nucleic acid binding, so these are the ones that regulate DNA in general, and the signaling molecules, then you got approximately a third. So that's the biggest category, when like, or meta category, if you want, of genes. The rest are mostly enzymes, structural components, or unclassified, which is of course by far the biggest. But that corresponds to actually different functions here. There are approximately 20,000 protein coding genes, and there are many more non-protein coding genes, such as the long non-coding RNAs. But about the long non-coding RNAs, there is serious doubt whether they do anything at all. Okay, they produce an RNA. This RNA is not translated, but they still produce an RNA that goes in the cytoplasm or that may stay in the nucleus. And of course, that RNA can fold, can have some enzymatic activities even, but in practice, it's only for a handful of these, hundreds of thousands of long non-coding RNAs, tens of thousands, that we have a function. Most of them, they just seem to be an accident of transcription. Still, they count as genes because they've been transcribed and they have a product, and there's much more of them than protein coding genes. Another part of the genome and of the genes are the mitochondrial genes. So the mitochondria, remember, that's this thing that looks like that with the digits that are like this. It's in the cytoplasm, and it's got DNA, like a plasmid, like a small plasmid of 16,000 base pairs that's inside the mitochondria and allows it to grow and reproduce. So mitochondria have 37 genes, including 22 tRNAs. So most of the genes are tRNAs, 2 rRNAs, so ribosomal RNA, and 13 protein coding genes. So these are all these genes, the 13 protein coding genes of the mitochondrial genome here. They're all involved in the function of the mitochondria. All these genes produce a protein that will go to the mitochondria for the oxidative phosphorylation. So here they're highlighted. These are the enzymes of the oxidative phosphorylation. So they will help produce ATP. So this is a fairly specialized genome, but it doesn't contain everything. Many mitochondrial proteins, actually most of them, are encoded in the nucleus. Okay, so all the genes that are in the mitochondria have something to do with the mitochondria, but there's still a lot more that are outside, that are in the genome, that produce the missing pieces. Part of these remaining 10% that are neither repetitive nor genes are pseudogenes. So pseudogenes are non-functional, they're mutated genes, and they can be transcribed if their promoter is functional, but they do not produce a functional protein. Okay, so a pseudogene is something that should be producing a protein, but that does not. That's kind of the definition, the loose definition of a pseudogene. There are approximately 20,000 pseudogenes in the human genome. That's the same number as the protein coding genes. So that's the mechanism by which you can have a pseudogene that's produced inside the human genome. They can be reverse transcribed again by the line 1 method. And after this, once they incorporate in the genome as a DNA, they can mutate and they lose the capacity to produce anything interesting. So most of the time, pseudogenes have no introns. That's one way that we can easily recognize them. And so back to one of the first questions that was asked about the human genome, well, it's still not finished. There are still gaps. So when you take that particular chromosome here, chromosome 1, and then you zoom in around that region, this is what you see here. Okay, you zoom out to the maximum and you go close to the centromere, and basically there's nothing. You look at it, there's no gene, there's nothing. So is there really nothing there? Or it's just that we don't know what there is. So there are gaps in the assembly. They're often located close to the centromeres. And last time I checked, there were 79 gaps in the human genome, in the most up-to-date version. And they represent 5% of the genome. So that's not really small, actually. 5% is non-negligible. So the human genome is not completely finished because of these 79 gaps. There's a question for you to think about, and I'm not going to answer it. It's just for you to start imagining. Why do you think that these regions are hard to sequence? You know, if you zoom in around the centromere, why do you think it could be so difficult to see what's happening there or to know what's there? But we're not alone. There are other species on this planet. And one species that's in particular quite interesting is our closest relative. So when we look at the family tree here, the phylogenetic tree of the human species, as I'm sure you know, chimpanzee is our closest relative. In reality, it's either chimpanzee or bonobo. There was a split between these two species after both of them separated from humans here. And when you look at the karyotypes of chimps versus ours, there are very, very few differences. There's only one actually, and I needed to look for it and to highlight it for you because it's otherwise very difficult to see. That diagram here on the right is the comparison of the pairs of chromosomes from chimp to human. And is that back there, here, you know, like 5 million years ago, something like that, our chromosome 2 were 2 chromosomes. And so in the chimps, it's still 2 different chromosomes, but in our lineage, they have fused together to create 1 chromosome. Like the 2 centromeres, that's where my tips of the fingers were, they fused and that created just 1 non-chromosome, which is now our chromosome number 2. And that did not happen in the lineage on the left, the one that gave chimpanzees and bonobos. So that's the major difference in terms of karyotype. The divergence with chimpanzees is approximately 4%. So there are 35 million single nucleotide differences. So SNPs, if you want, except that SNPs is not applicable. Okay, SNP is within a species, it's not between species. So that's approximately 1 difference every 100 base pairs. Compare that to 1 difference every 1,000 base pairs when we compare 2 humans together. And there are approximately 90 million base pairs of insertions and deletions, which together represent 2.8% of differences. So these are the nucleotides that are either present in human but not present in chimp, or the reverse, present in chimp but not present in humans. And that's the bulk of the diversity. It's not single nucleotide differences, it's large insertions and deletions. But still, we're not just humans and chimpanzees. And when I said that the human genome is the most important genome on the planet, well, that's what everybody would have said before 2020. And somewhere around early 2020, everybody said, no, there's another genome that's kind of more important than the human genome. And that's, of course, the hated genome of SARS-CoV-2. So I wanted to give you a little bit of a heads up of what genomics has already been able to tell us about SARS-CoV-2, so that you can kind of start to grasp a little bit what's the idea of genomics and how that can be useful to understand, or at least to have a, you know, a prism to look at the events that are happening in the world right now. So the SARS-CoV-2 appeared in 2019. That's the one that causes COVID-19, of course. And its genome was sequenced in January 2020 in Wuhan. The genome is made of a single-stranded RNA, and it looks like that. So that's here a picture of the SARS-CoV-2 genome. And you can see here that there is approximately 30,000 nucleotides of the genome. So here, the picture I got is inaccurate. It says base pairs, but that's not base pairs. It's actually nucleotides because that's a single RNA that constitutes the genome. So don't mind that. That should be actually nucleotides, not base pairs. And there should be only 1 line because this is a single-stranded genome. As you can see, the genes are indicated here, ORF1A, ORF1B, S, E, M, N, and the so-called accessory factors in green that are there. An important consideration is the color of these boxes. The darker the color and the more the protein is similar to the SARS-CoV virus, the one that appeared actually in Toronto in the beginning. I can't remember the years. I think it was around 2004 or 5, something like that, where this virus appeared. And it's very much related to the SARS-CoV-2 genome. Like these 2 viruses share a common ancestor that was not too long ago in the past. And you can see that most of the proteins that are here have a dark color, which means that it's practically the same in SARS-CoV, the SARS-CoV-1. The major differences are here, 3B and 8 here. These accessory factors are quite different. Unfortunately, many of these proteins here have an unknown function, and 10, sorry, I forgot. Many of these proteins here have an unknown function. So it's not completely clear, for instance, what are the main differences between SARS-CoV-2 and SARS-CoV. Like, why is one so deadly? SARS-CoV is an absolute nightmare. Like the death rate is very high compared to SARS-CoV-2. I think it's not completely clear exactly why that is. So there are other proteins like the S, E, M, and N, and there are the structural proteins. So these are the ones that kind of facilitate the life of the virus. Some of them are masquerading the virus away from the immune system so that it can escape, it remains invisible. And these ones are really, you know, creating the physical object that is transmitted between cells, the structural proteins. So the protein S is the spike, E and M form the capsid that contains the nucleic acid, and N is a packaging protein for the RNA that goes inside the capsid. On top of this, you have the envelope, that is like the lipid bilayer that goes around the capsid that it steals from the human cell that it uses to infect. And it's from the envelope that the spikes are pointing. So we're going to review very rapidly the replication of SARS-CoV-2. I'm sure that you guys know this already, and I'm sure that many of you actually know more about SARS-CoV-2 than I do. But it's just to put everybody on the same page, and also to just highlight, the fact that we live today and that these things are happening today in a world that already is proficient with genomics. What are the changes in the way the epidemic pans out? So here the picture is kind of too small to be seen. And so I'm going to zoom in on different pieces so that you can see the whole life cycle of SARS-CoV-2 bit by bit. So we start here on the top left corner, and I've magnified it here so that you can read it more comfortably. So the first thing is that the spike protein, the one that's represented here in pinkish with these things that protrude out of the envelope of the capsid and also the envelope. The spike protein of SARS-CoV-2 binds ACE2, the receptor. So the virus then enters the cell and releases its genome. So here, this is a representation of the target cell, the one that would contain, for example, that protein ACE2, and the spike binds that efficiently. That's what's represented here. And that's what allows the virus to kind of get absorbed inside the cell, like either through that route or through that route. What happens anyway is that the binding of ACE2 to the spike, it triggers a change of conformation of the spike that then allows the membrane here to open and to fuse with the plasma membrane of the cell. And that's what allows the virus to release its genome inside the cytoplasm of the cell. So you have to remember that the genome of SARS-CoV-2 is RNA. It's already, it can be translated directly. It doesn't need to be transcribed. So that's already an RNA inside the cytoplasm, ready to make new proteins. So the RNA is now inside the cytoplasm of the cell, and it can already produce the proteins that will start the replication cycle. The cycle continues here in this part of the picture, and it's been kind of magnified here so that you can see it better. So one thing that must happen is that because the genome is single-strand RNA, it needs to make more copies of that single-strand RNA. And the way it works is that it's first making a negative copy of the single-strand RNA. That's it's called the plus strand, the one that travels between cells, and it's going to make the minus negative strand using a protein that's called an RNA-dependent polymerase. So the RNA genome is transcribed via an RNA-dependent RNA polymerase, or DRP. That's the one that's indicated here, to create a double-stranded RNA. Okay, that's kind of weird to say that you produce another copy by transcription, but that's why it is. Okay, that's an RNA-dependent transcription, something that uses RNA as a template to produce RNA. More copies of the genome are produced by transcription. This also produces RNAs of different sizes that are translated by the ribosome to produce viral proteins. Okay, so you see here these double-stranded RNA that are produced here. Then you can also use RDRP again to produce now this time the plus strand. And depending on where you start transcription, you have RNAs of different sizes. So these are kind of normal mRNAs. If you want, they have a cap, they have a poly A tail, and you produce a cascade of RNAs of different sizes here. And if you remember the structure of the genome that looks like that, it means that you're going to produce more of the proteins that are on the right, the one that are on the 3' side of the virus. So you produce more of the structural proteins like the spike, EM, and N that are on the right than the protein on the left. And the reason is just because you produce more RNAs that are short, that are biased to the right, to the 3' end, then you produce complete RNAs with everything. Okay, so these are RNAs. They look like normal mRNAs, so they can be translated. It's no problem. Like the ribosome comes and then produces the viral proteins that are used to complete the replication cycle. I think I made a mistake here. So I said that E and M form the capsid. I think they're in the envelope, not in the capsid. So the final part of the replication cycle is here on the top right. So when enough proteins are made, SARS-CoV-2 forms new capsids and envelopes, and it escapes the cell through the Golgi. Okay, it never goes in the cytoplasm, like the translation all happens, sorry, it never goes in the nucleus. The virus just injects its genome in RNA form in the cytoplasm that's ready to be translated. You have this RDRP mechanism that produces new viral copies, also new RNAs that are used only for translation. And then once you have enough proteins like that, you can start forming the capsid, packages the RNA genome inside, and then that package here is kind of going through the Golgi and then exported outside the cell, and the replication cycle is complete. So that produces new particles that are ready to infect new cells that have the ACE2 receptor on their surface. So I'm sure that this is some things that you all knew. There was so much was said about SARS-CoV-2 already, and there's nothing special about this virus. This is just a standard replication cycle for any virus. The highlights of the coronaviruses is that they're RNA viruses. They never have a DNA form, and that they never really enter the nucleus. All these things happens only in the cytoplasm. So one thing that's particularly interesting is the spike protein. So this is here an electron micrograph picture of SARS-CoV-2 that was taken at the beginning of the epidemic. And you can see that in electron microscope, it looks like that. It's got this characteristic form here when you see the envelope, and then you see these spikes that are going out of the envelope like this. And this is why it's called the spike protein, because it really looks like spike when you look at them. This is a zoom in here on the spike with a crazy magnification with the electron microscope. You can see that the scale here is really, really tiny. We're watching things that are very, very small here. So the bar is 50 nanometers, and this is 10 nanometers. So the spike is approximately 10 nanometers here, protruding outside the envelope. So the spike or S protein of SARS-CoV-2 points out of the envelope and forms the characteristic crown of coronaviruses. It even gives the name to the coronavirus. The corona, or the crown, really comes from the fact that they all have these spikes pointing out of the sphere that's like that. It is the main protein that interacts with the receptor on the cell surface, ACE2, allowing the virus to enter. So at the beginning of the epidemic, people were able to make a 3D rendering of the spike protein. And one thing is that depending on whether it interacts with ACE2 and the co-receptor, its structure is very different. So it looks like this when this is in its kind of native normal form, when it goes out of the cell. And here that represents actually a trimer of the protein. And once it's been interacting with ACE2, that each trimer has interacted with it, there is a cleavage that changed completely the structure of the protein. And you can see that it looks nothing like it used to. So that kind of makes it a little bit like a thrust, something that goes inside the cell. And that's what's allowing the virus, if you want, to open the membrane and then to kind of fuse with the membrane. That's the post-fusion here, the so-called fusion peptide that usually goes inside the membrane of the cell in order to kind of open it like a curtain. And so why am I telling you that? And why am I linking that to genomics? Is because there's an RNA that got a lot of attention, and that's called mRNA-1273. And mRNA-1273 is the basis for the Pfizer vaccine. And I know that many of you have received that vaccine. I have it. I got 2 vaccines. I got vaccinated 3 times because in Austria we were a little bit ahead of the curve. And so that's why I wanted to talk to you about this, because I know that this is an experience that many of you have had. And this is something that was possible only through the fact that genomics has really come of age as we speak. So production started 5 days after the publication of the SARS-CoV genome. So this is the information that you can read on the article that is there. But this is in a sense that's something that's unheard of. Some people understandably are a little bit scared that things went too fast for the production of this vaccine. But one has to understand that going that fast was just not possible, let's say 5 years ago, or something like this. The other thing to understand is that it doesn't come from nowhere. People had been investing very heavily on coronaviruses since 2017. So in particular, Pfizer and other people around the world were already studying the spike protein because of the SARS that came around 2005. So people realized that there was a danger, and so they started a program of readiness for epidemic and they had already worked many years on that protein before COVID-19 appeared. And so one thing that they developed is from the MERS virus, the Middle East Respiratory SARS syndrome, sorry, Middle East Respiratory Syndrome. They had developed this technique to stabilize the spike protein using these mutations that are called 2P. So these are two proteins that they put around there, and that stabilizes the protein in its native form, which can be used then for producing a vaccine. So you can see that in 2017, this was already done. People were already able to produce these proteins here through this mRNA. And when the problem started in 2019 and then 2020, as soon as the virus was sequenced in January 2020, the next day, they were able to design an mRNA that were producing that proteins in vitro because they already knew how to stabilize it, so it will actually create potent antibodies. So they designed the mRNA on January 11, and they produced it on January 15 onward. It was already pretty much the same as the one that you received today, or a couple of months ago, I mean. And so when you think about this, this is possible only because people have had this kind of look at readiness for epidemic through the genomic prism. The clinical trial started kind of a couple of months after that, and of course most of the time that was spent was on checking the safety, etc., and the efficiency. But in terms of production, what you have to understand is that really it's because we live in a genomic era that we're able on day one after the publication of the virus to start the design, and on day 5 to start the production. This is absolutely impossible to imagine any of this happening a couple of years ago. So that's the story of mRNA-1273, showing basically that when we live in the genomics era, we can do just more than when we live in another era. Of course, that's not the end of the story. We know that enter Omicron. So this recent variant kind of changes the deal a little bit. What you can see here is the evolution of the SARS-CoV-2 from the beginning of the epidemic at the end of 2019 till now. And what you see on the y-axis is the number of differences compared to the original Wuhan strain that is there. The BA.1 or Omicron variant was first sequenced in November 2021. Its spike protein diverges substantially from the original Wuhan SARS-CoV-2 strain. So that's the number of mutations only on the spike. And as you can see in the phylogenetic tree here, it's a bit of a mystery where it comes from because it doesn't actually branch with the other variants. Like the other ones, we understand they come from each other, like first beta, then alpha, then delta. But Omicron comes from somewhere else. You can see that it branches actually here, like from somewhere in the early 2020 or mid 2020. So it seems that most of the evolution of the Omicron has been underground. It's not been followed at all. And it came a little bit out of nowhere. I think I'd like to see a little bit more information about where this is coming from, because as you can see from that graph here, it's not really related to the strains that were already out there. It comes from somewhere else. And of course, the reason why we're having this class today online is because of Omicron, nothing else. So the mutations on the spike protein of Omicron are on the surface of the trimer rather than on the inside. So what you can see here is a rotation model of the spike protein of Omicron, and every letter that you see here is a mutation compared to the original Wuhan strain. And here, this is a part that is actually from the cell. It's not a part from the virus. That's probably the ACE2 receptor. But just have a look at where the mutations are here. They're on the outside. The antibodies that recognize the original spike protein, that is to say the ones that were produced by mRNA-1273, do not recognize the spike protein of Omicron very well because of those differences. I think you understand exactly what happens. Like the RNA was producing another spike that was actually very good at producing antibodies, but Omicron doesn't really have exactly the same spike. It's got mutations like all over the place, which means that the antibodies that you've got are not really good at recognizing that particular spike protein. And so the virus goes through the immune defense without being recognized. So that's what I wanted to share with you, just to make sure we're on the same page and we understand a little bit at the intellectual level why we're having this conversation online instead of being in the same class as was originally planned. So that's it. The class is finished for today. So the key point to remember are that protein coding DNA is a tiny fraction of our genome, but it has a large potential for regulation and signaling. Okay, like our genome has a large potential for regulation and signaling. That's highlighted by the fact that 1/3 of our genes are involved in signaling. Also, our genome is not just our own. It is a home to many selfish DNA elements. Actually, there are much more selfish DNA elements than our cells in our genome, such as transposons and viruses. And that's it for today. So now it's in 1 minute, the quiz will open and you'll be able to take the quiz. That's kind of a way to jog your memory. My advice for you is read the questions nowish, maybe tomorrow, something like this. You have 6 days to answer them. And I really suggest that you at least read the questions, take your time to think about it, but don't read them in the last 5 minutes. Oh, sorry about this. I turn on the light again. Anyway, the class is finished. So I'm going to stop the recording, stop the sharing, but I'll stay online for those who have questions.